So welcome back. So at the end of the last one, you were probably thinking to yourself, so we were doing structures of the ear, but all we did was external ear and middle ear, but she had said there was an inner ear, but I don't think we covered it. And you're right, because we didn't do the inner ear. And that's because the inner ear has structures both for hearing and for equilibrium. So in this part, we're going to talk only about the structures for hearing, because it's very common for students to be a little bit confused. I don't understand how you went from this picture to that picture. So let's go ahead and get started. So this will involve that cochlea, that snail-like structure inside of which we have the cochlear duct. And so previously we've talked about it being a little circle inside a big circle, but in the cochlea, I want you to realize it's not sitting right there in the middle. That inner duct structure is actually off to the side, and it will divide with membranes the entire cochlea basically into 3 different areas rather than one outer area and one inner area. So if we take the cochlea and uncoil it, as in the upper left, and we would make a cross section through it here, it would give us this picture, which is the cross section of the cochlea. And then if we took the boxed area and blew it up, it would give us this bottom right picture of a structure known as the spiral organ. And then if we were to look at the upper part of this spiral organ, and this is a scanning electron micrograph, that's what the view is from above over. So we're going to get to the last part, but we're going to start with the first part. So inside the cochlea, so what happens is between the middle ear and the inner ear, we have those 2 membranes at the oval window and the round window. And what they do is on the middle ear side, we have air, but on the inner ear side, we have fluid. So we have to change or transduce those air vibrations into fluid vibrations. So everything on this side is going to have fluid. So when we did that cross section in the previous slide, okay, and because I said the cochlear duct is pushed off to the side, it gave us an upper area and a lower area, and the middle area was where the cochlear duct is. So here is the cochlear duct. It's kind of this triangular thing in the middle, and you can always tell the orientation because this structure in the box sits on the bottom, sits on the floor of the cochlear duct. Okay. The upper chamber is known as the scala vestibuli, and the lower chamber is known as the scala tympani. And the easiest way I know to remember is your vest, as in vestibuli, is above your pants, as in tympani. So looking at these 3 areas inside the cochlea, there are dividing structures, there are membranes. So between the scala vestibuli, whoops, I went the wrong direction, my apologies, okay. Between the scala vestibuli and the cochlear duct, there is a membrane. And between the cochlear duct and the scala tympani, there is another membrane. And the structure in the box, which is the spiral organ because it goes in a spiral, also known as the organ of Corti, as in the guy who named it, there is a 3rd membrane in the top of the spiral organ, and that is this structure right here. So the membrane that's just below the scala vestibuli that makes the floor of the scala vestibuli is called the vestibular membrane. And the membrane that makes the floor of the cochlear duct is called the basilar membrane. And the 3rd membrane, the one that's at the top of the spiral organ, is called the tectorial membrane. So let's look at that spiral organ a little closer. So we're taking that boxed in area and blowing it up. The spiral organ sits right on that basilar membrane, okay? And And at the top part of it, we have that tectorial membrane, which is attached. And you notice on this side is the side that it is attached on. So the side closer to the attachments, we call those cells the inner hair cells. We call them hair cells because it looks like a cell that has like a marine fruit bud on top of it. And then the cells further away from the point of attachment, they are called the outer hair cells. And it's the outer hair cells we're going to be talking about right now, because what they do is they amplify low sounds, okay? And the inner hair cells we're going to spend a lot of time on. The last thing on this picture is if you look at the basilar aspect of each of the hair cells, you will notice there's dendrites going to the cochlear nerve. So the cochlear nerve is the nerve that gives the sensory input from the entire cochlear duct. Now, the hairs on the top of the hair cells, what they are is they're very stiff microvilli, but we call them stereocilia. And among the stereocilia, there are going to be more than 50 of them for every hair, and they're pretty stiff. And then there's going to be the one that's the longest. And the one that's the longest is called the kinocilium. So it gets a special name. And the reason for that is when we are talking movement of the stereocilia, we are going to be talking as, you know, like what direction is that kinocilium? Are the other stereocilia moving towards it or away from it, or what is happening? So let's look at these inner hair cells. So remember, we are back over here close to the point of attachment. So here is the inner hair cells with the stereocilia. Here is the longest one, so that's the kinocilium. And there is the tectorial membrane above it. We have endolymph in the cochlear duct above it, and the basilar membrane down below it, and there's the cochlear branch of the vestibular cochlear nerve. So what these inner hair cells are going to do is they're going to convert sound energy into an electrical signal. So first we need to talk about what kind of structures we have going on here. So this is a close-up of the tops of the stereocilium. And on the top of each one, do you see this little blue dot? This little blue dot represents an ion channel right here. And the ion channel of a shorter stereocilium is connected to its taller neighbor by a protein called a tip link. So it links the tip of a short stereocilium to its next taller neighbor, all the way up, all the way up, all the way up, all the way up to the kinocilium. When these ion channels will open, that is going to allow potassium to enter the hair cell, thereby changing the polarity of that hair cell. So we'll talk about that later. And that's it for structures of the inner ear. So come back and let's learn how hearing happens.